Hello, everyone. Welcome to Introduction to Biosciences. Hi, I'm Anna, a member of the UCL IGEM 2020 team. So our team has 8 undergraduate members and 3 supervisors. We'll all be contributing to our 3 webinar sessions. So last one, our webinar 1 session was happening a week ago, and then it was focusing on subjects such as molecular bio, natural sciences, and immunology. Yes, here's the overview of our webinar sessions. And a YouTube channel we made included the cast for our webinar 1. So later on, we will send out the link to see that the webinar 1 contents. And today we're going to focus on biochemistry. And next week, we're going to talk about biochemical engineering. So looking forward to that.
Greetings, everyone. My name is Stefan. Me and Pedro introduced molecular biology last webinar session. I am a third-year molecular biology, a member of the IGEM team, and it's great to have you.
Hi, guys. This is Pedro here. As Stefan said, I was also part of the molecular biology webinar introduction. So that's also something that's on the UCL IGEM 2020 YouTube channel if you weren't able to make it to the last one. And yeah, I'm part of the IGEM team. I'm a second-year molecular biology student, and I can't wait to see what we can achieve this year. Thanks for coming, guys.
Hi, everyone. My name is Juliet. I'm a third-year natural sciences student at UCL, and I was part of the team last week to introduce natural sciences to you guys. And I hope you enjoyed it, and I hope you enjoyed today.
Our supervisor, Professor Kent, is not able to make it here today, but he was introducing immunology last week, so please go to our YouTube channel to see more about immunology and COVID-19. And now let's meet the speakers for today's webinar session.
Hello, everyone. My name is Daniel. I'm a third-year biochemistry student, and I decided to join IGEM because of its multidisciplinary aspect. So I like when people with different backgrounds work together to achieve a particular goal.
Hey guys, thanks Danny. Yeah, I'm Oli. I'm also a third-year biochemistry student. I'll also be one of the speakers today. I'm in the same class as Danny, so we hope that we can give you some good talk and some good guidance today about some of the stuff we've learned through our short studies so far.
Hi guys, I'm Alex. I'm doing a PhD here at UCL and helping advise the IGEM team this year. I did biochemistry as part of my undergraduate, and I'll be talking to you a bit later today as well.
So now we are meeting the speakers for webinar 3, which is happening next week, talking about biochemical engineering. Hi, I'm Anna, first-year undergraduate student studying biochemical engineering at UCL. Besides biology, I'm also passionate about coding and robotics. I joined IGEM because I wanted to learn how programming could facilitate SynBio research. And looking forward to seeing you all in webinar 3.
Hi everyone, I'm Laide. I'm a second-year biochemical engineering student. I applied to IGEM because I wanted to work on an interdisciplinary project where I could develop my computational skills and also learn more about synthetic biology.
Hello everyone, I'm Lee, a second-year student at UCL, majoring in bioprocessing of new medicines with business and management. I'm interested in how to commercialize biological research, improve biopharmaceutical manufacturing and marketing, and any other bio-related stuff. If you are interested, I'm happy to talk with you guys more in session 3.
Thanks for introducing yourselves, my team members. And my supervisor, Stephanie, is going to join us in webinar 3 to talk about biochemical engineering. So now let's move on to the next slide. In our 3 webinar sessions, we're going to go through the bio majors that we are studying right now, or the bio majors that our supervisor studied before. So those include biochemistry, molecular bio, biotech, biochem engineering, immunology, and natural sciences subjects such as medical physics and neuroscience. So within our 3 sessions over the summer, we're going to look into the applications as well as career potentials of these majors, and we're also going to share our journeys into these majors. So now I'm going to hand it over to our webinar 2 speakers to talk about their major, biochemistry.
Okay, so in biochemistry, there are several core modules that we have to take, and they can be classified into different levels, depending on the level of detail, of course. So you can, there is the molecular level, where it's the smallest level and the more detailed one, where we look at signaling and metabolic pathways, such as the Krebs cycle. We look at protein structure and function, such as hemoglobin here, which is a protein in charge of carrying oxygen to all parts of the body. But also we look at how the DNA can be modified through molecular biology to achieve different functions. At the cellular level, we have immunology and infection as electives. At the larger level, we have systems for engineering level, where you're actually modifying a whole organism to make it do something new, a new function. So a definition of biochemistry would be bringing chemistry to life. So it combines the physiochemical processes inside a cell, and those physiochemical processes allow the cell to be alive, essentially. On the left, we see a picture of an analogy of the cell as a factory where nutrients come in, and the nucleus is in charge of the DNA replication, transcription, and translation. But it also needs to deal with metabolic waste. And these processes need to be highly regulated at the molecular level, indeed. If they're not, then the cell cycle could experience some abnormalities, which can lead to disease such as cancer, for example. The applications of biochemistry are very broad. You can go from studying protein structure. For example, on the right, we have a picture of a ribosome and a nascent polypeptide chain coming out. For this, we use cryo-EM to visualize the rigid parts of proteins, whereas for more flexible parts that are tumbling in solution, we use a technique known as NMR. And we often use these structural techniques to visualize biochemical processes inside the cell. Another application would be metabolic engineering, so like tweaking metabolic pathways inside the cell to make it produce a particular product. Obviously, there are many applications in healthcare and drug discovery, as well as immunology, things like vaccine. Then you also have the more bioengineering approach, the biotechnology, where you have these startups that are specialized in making specific compounds and engineering cells in a particular way. But also another aspect of biochemistry is the computational side and the data science and analytics. You can imagine this as a very dense and complex database, whereby the role of these scientists is to decipher the meaning and actually classify the information to understand it a bit better. The careers you can take with a biochemistry degree are also quite broad. You can focus on you can be a biochemical process developer in industry. You can research proteins and how they relate to disease. For this, I'll give an example. This here is a protein folding funnel. You probably know that the proteins need to fold correctly in order for them to have the correct function, right? So as they come out of the ribosome, they fold spontaneously. And it's quite impressive that they do this, actually, with the most thermodynamically favorable state being the one with lowest energy. However, sometimes things go wrong and the proteins can aggregate as they fold, forming intermediates. And then you have pathology. An example of this is the beta protein, which gets stuck in an intermediate state and never reaches the native state. This actually causes Alzheimer's. And you can see the difference between a healthy brain and advanced brain due to protein aggregation. Obviously, drug discovery and biotechnology industry, they are key. They're good there are applications of a biochemistry degree, but also sports consulting and supplementation, because the metabolism of your body is highly connected to what you eat, obviously the amount of sport that you do. Now we're going to talk about our journeys in particular, and we're going to give specific examples. So, surprise, originally I was not a biochemist, I was a biologist. I didn't know what to choose in the beginning, so I decided to go broad. And then within a week, I already knew the organism level, like animals and ecosystems, was too big to answer my questions. So I knew that I had to look more into the detail of these processes to understand the origin of life, indeed. And we're talking about DNA, the process of DNA replication, transcription, translation. Once you reach the protein level, these proteins actually perform function. And this was able to allow me to understand more the origin of life by looking at the function of specific proteins. Things that I like and dislike about my course. Well, I love the molecular regulation of cells. I think it's fascinating how these tiny little things have molecular machines that regulate the processes and allow them to be alive. So essentially you have inert chemical reactions. They're not alive, but they're happening. And somehow when all of these reactions are combined together, they allow life to happen, and cells are alive. Another thing that I like is the genetic engineering of cells, and I'll give you a real example of that. So in our immune system, we have normal T cells, which cannot recognize cancer cells effectively. So because the cancer cell expresses inhibitory molecules in the surface and it hides from the immune system. We can use genetic engineering to engineer the normal T cells, and actually you can extract these T cells from the patients and actually train them, if you wish to say it like this, we train them by to recognize the cancer cell, and this is known as CAR T cell therapy. But also we can another thing that I like is how these processes lead to disease. So this is the ROS signaling pathway, whereby ROS is a protein that is located at the center. Obviously, it regulates this pathway, which allows cell division, survival, and is essential for cells to be alive. However, in cancer, ROS is often mutated, and this leads to over-proliferation and uncontrolled cell division, causing cancer. And I'll give you an example of how that can be addressed and how we are trying to target ROS at the moment in the clinic. Things that I dislike about biochemistry is that cellular regulation is often complex, and there are many metabolic and signaling pathways that you have to learn, and they're a bit of a pain. So an example of how to target ROS, the protein that we mentioned previously, is by combining biology and chemistry. So, but this paper came out in 2020, so this year. It's a novel way to target ROS because up until now, people have been making chemical inhibitors that bind to this cysteine 12 mutant form of ROS, which causes cancer. However, this wasn't efficient because ROS is heavily mutated, undergoes lots of mutations, and then it acquires resistance to the chemical inhibitor. However, when you link this approach to biology, you can make a bivalent drug, which has the chemical inhibitor and the E3 ligase recognition site, and this 3 ligase recognition site allows for an enzyme to recognize this site and post-translationally modify it in ROS. It incorporates an ubiquitin, which is a molecule that targets proteins to the proteasome, which is a molecular machine that allows degradation of unwanted proteins. So in this way, ROS can be targeted with the chemical inhibitor and directed for proteasomal degradation. You'll see this coming up soon in the clinics, I'm sure. Another aspect that I love about biochemistry is the bioengineering part. So biotechnology and synthetic biology, where you take something that cannot do the function that you want, but you can engineer it at the genetic level to make it to acquire that function. So, for example, we have this plastic bottle, which is made of the polymer PET. It turns out that an organism was found, not E. coli, a different organism was found to have an enzyme known as peptidase that is capable of degrading this plastic bottle. So E. coli, which cannot produce peptidase, can be engineered to produce it. Also, you can link it to a GFP, which is a molecule that we discussed in the previous webinar from Okoria, Victoria, and allows the peptidase to fluoresce. So in this way, you can localize peptidase inside the E. coli, and that's why I do the E. coli in this different green color. So you can see how here you're engineering E. coli, which cannot degrade PET. You're engineering it at the DNA level to produce this enzyme, and then E. coli is capable of degrading PET. You can also engineer proteins. You don't have to always engineer organisms at the genetic level. You can also engineer proteins. An example is MATB, which was discussed in the previous webinar, whereby it is an enzyme capable of binding ATP. This causes a conformational change. And we can take advantage by engineering the protein. We can engineer two fluorophores, and due to the conformational change, we get a change in fluorescence because the fluorophores come apart. So here we have the N-terminal lobe and the C-terminal lobe, and the two fluorophores shown in green. And the fact that you have one fluorophore engineer in the blue domain and the other one in the red domain allows them to come apart when it binds ATP in the center between the red and the blue domain. Okay, finally, my research interest, I would say my main research interest at the moment is the production of compounds. So in my internship, my last internship, I actually targeted the cytochrome P450 enzyme, which allowed this pest. It's the diamondback moth, which is causing, which eats crops in North Africa. So my job over the summer was to develop a compound that would allow selective targeting of this enzyme that was causing the metabolic resistance to pesticides. And this compound had a chemical inhibitor part as well as a biotin. The chemical inhibitor would bind specifically to the P450 enzyme, and then a linker to a biotin molecule, which is a vitamin. The biotin molecule allowed to pull out the protein from the diamondback moth. And once we pulled it out, we were able to study the protein further and understand why it acquired resistance. And then after you understand how it acquired resistance, you can develop a different pesticide. And in this way, you can target this pest. However, I realized that chemical synthesis is very nasty in some cases, and it requires multiple steps, with most of the chemicals not being environmentally friendly. And I realized that you can do the same thing using a bacterial host. So you can take E. coli, for example. We like to use E. coli because it's simple, and you can give it reactions from different organisms. So you can take a reaction from the red bacteria, another one from the blue, and another one from the purple. And you can engineer E. coli to make a compound, which would not be able to be produced, which wouldn't produce naturally, essentially. So my career plan is to do a master's in synthetic biology and then do a PhD in engineering enzymatic cascades. If you're wondering what enzymatic cascades are, it's basically what I just explained in the previous slide, where you take enzymes from different organisms, put them in a single host, and you get that host to do the reactions you want. This paper is an example of the most efficient CO2 fixation cycle made so far, and it's fully synthetic. It's much more efficient than the natural Calvin cycle found in plants. And what they did here is they took the core structure of a carbon fixation cycle, and then in the dashed arrows, A, B, C, they incorporated modules, module A, module B, and module C, from different organisms with reactions that were much more efficient. And this allowed the creation of a fully synthetic cycle using reactions from all domains of life. So bacteria, eukaryotes, and archaea. And you're expressing all those reactions into a single organism, which I think is quite fascinating. So depending on the level of detail that you want, you can, I started in biology, I didn't like it. You might like it, but I didn't. I found it too big and it didn't answer my question. So I decided to go more for the molecular detail. But remember that you always need passion. And I'll pass it over to Oli.
Right, thanks, Danny. That was really cool. Thanks everyone for joining in. So, as I mentioned before, I'm also a biochemistry student in the same year as Danny. And some of the stuff I want to talk about, obviously it applies to biochemistry specifically, but I just want to give you guys some guidance that really helped me when I was trying to figure out what I wanted to do with my life, my career and all that kind of stuff. So first of all, if you're here, I assume that you're interested in technical fields in that sense, particularly biosciences. So something that really helped me out just to try to figure out what I want to do with my life is to have a little reflection with yourself and figure out, first of all, would you rather try to understand what's there? So do you gravitate towards science, or would you rather try to use what's there to solve problems, right? And then in that case, you more or less gravitate towards engineering. Obviously, neither of these means that if you choose one, you don't choose the other one. This is not black or white. More or less the opposite. Science and engineering feed each other all the way through. But it's good to more or less know where you want to sit in that spectrum. It'll give you some really useful long-term guidance. So just to get this message across, I drew this diagram on the right, where basically you have science on one side that is able to inform engineering in order to generate new inventions with the stuff that you learn from science about understanding what's there, basically. Whereas on the other side, engineering is able to provide the technology with these new inventions that is able to push science further to understand what's there even more. You know what I mean? So they just feed into each other and they keep growing together. Danny? Sorry, we're just, my friend has to press on the slide. So in terms of what I like and what I dislike about the course, I think the coolest thing about studying biochemistry is that you are able to understand biology through all its layers of complexity. And what I mean by that is that you're able to see what are the underlying chemical reactions behind all biology and how that complexity arises into building a whole organism. From that whole organism, you can go a step further and understand how organisms deal with each other in ecosystems and how they interact. And to me, just being able to understand all of the complexity of biology through its layers, obviously I'm really far from that, but it's a really, really cool endeavor. And it's something that I find very fulfilling and fascinating. In terms of something that I think I struggled with the course is that, Danny? No worries. So something that I think I struggled with in the course was the fact that this thing being about either concept-oriented or detail-oriented. What I mean by that is that in my case, I think I'm quite concept-oriented in the sense that my brain doesn't really obsess with the details, and it tries to like some way to try and see the bigger picture to understand the concepts behind this stuff. Whereas other people might be more detail-oriented and they obsess with the details and they just want to go further and further with those questions, right? So just to give you an example of this, Danny, obviously this is not black or white. It doesn't mean that one person is only one of those two, but Danny, I would say, is more detail-oriented than me, right? So as you could see in the previous slides, he was very interested in looking at the particular pathways that the particular proteins that are involved in one pathway, and particularly the structure and how that leads to the function of each of those proteins in that pathway, right? Where if you are like that, if you're obsessed with the details, right, and obviously you will realize that at some point through your studies, this biochemistry is definitely a career where you're going to thrive because you will get an insane amount of detail. If you're a bit concept oriented, I feel like the detail can scare you off a little bit, and definitely did for me. But if you're able to learn how to deal with the detail and take advantage of it, it's also ridiculously rewarding. It will be super helpful to understand the higher level concepts. So in terms of like just some kind of corny goal that I would like to do with all of this stuff, it's quite ambitious and vague, but something that I would like to do throughout that, some driving force towards my career will be trying to understand the complexity of biology and then take advantage of the complexity to solve problems. And I just feel like that's a cool driving force to have at the back of my head whenever I'm trying to push forward, basically. So in terms of how to achieve that goal, the main thing that I'm interested in right now is known as systems biology. And systems biology is basically trying to understand organisms at the system level, right? So you're trying to understand how all of the pieces inside of an organism interact with each other and are regulated with each other in order to make up an organism, right? So a good metaphor for it is that usually you tend to like pick up all the pieces in order to understand them individually, whereas systems biology tries to put them together to understand the whole process and the whole system. In case you're a little bit like me and get distracted in these kind of talks, I just left a little diagram on the side for you to see more or less a metaphor about a cycle about what systems biology is. Then one thing that definitely is super useful for systems biology is that, as I mentioned, biology can be really complex, and you can't store it all in your brain. So it's ridiculously useful to take advantage of computing power and understand a little bit of computing, just to being able to model all this stuff and being able to understand the complexity through computation. And just a little hiatus here, a little side note, just for you all. If you think computational biology might be something that scares you off, or it's not super interesting, I felt the same. I didn't think computational biology was something that would be super interesting. I never gravitated towards computers. But I just want to say that as you start your undergraduate studies, you're basically a sponge, and there's no better time in your life for you to try new stuff and try learning new things. And when I first was confronted by coding, I was like, this is probably not something that I'm going to enjoy. It ended up being super exciting, and it was like learning a new language and super useful. So just be open-minded, and your undergraduate studies is the best period for you to try things out. In terms of so my end goal necessarily is not understanding the whole complexity of biology, although that's ridiculously hard. It's more than that, it's making use of that complexity in order to fix problems, right? So similar to the synthetic biology approaches that Danny was talking about before, with engineering metabolic pathways and that kind of stuff, right? So the end goal will be to learn how to properly engineer biology to solve problems. And obviously, complexity can be scary, but the complexity of biology actually, at the end of the day, is something really interesting, because the more complex the system can be, the more potential it has to solve problems. And appreciating that and acknowledging that is something that I found to be very fulfilling. Here on the side, I've just led a little picture of a treehouse, just because this was one of the first ideas that was introduced to me in terms of what synthetic biology is. And it was that, imagine if you were able to know and understand biology so much that instead of having to build a house on a tree, you could just plant a seed that you had engineered in order to make the tree grow into a house. So growing into a house instead of having to build a house in a tree. That was just something that's very science fiction right now, but it's a cool idea about how we can take advantage of complexity to solve problems. In terms of the stuff that I want to do, it's not going to be necessarily in this order. I mean, the masters will happen in this order. I'm going to be doing the same masters as Danny in systems and synthetic biology. And in terms of the stuff that I want to do after, it doesn't necessarily have to be in this order, but I would like to do some kind of PhD just to build up my expertise and my ability to use computers to understand biology. And then I would also like to work out in the outside world a little bit, just to understand how does that work. Because obviously, being a student, you're still quite shielded from how everything else works around the world, right? So here on the side, I mean, the end goal after that actually will be to develop the expertise necessary to solve some problems myself and enter into some kind of startup. That will be the end goal. And here on the side, I've just left concept that I think is quite corny. I found it quite corny at the start when I was first introduced to it. But it's something that actually really helped me in terms of understanding what I want to do with my life. Just because I feel like life can be really confusing, particularly if you're a kid that's growing up. And this concept just really helped me simplify what I want to do in my life. So it's called Ikigai. It's a Japanese concept, and it stands for reason for being, I think, reason for leaving. I'm not bilingual by any chance, but it's basically a mixture of four things. It's basically having not necessarily what you're good at, but what you can work at to get good at, right? What you love, so what kind of passions you have, what can sustain you, so what you can be paid for, and then what the world needs. And if you mix all of those, I feel like you can get a quite fulfilling life, at least in my opinion. And then just to wrap it up, you have probably realized I tend to overthink stuff quite a bit. So something that's really helped me out in terms of figuring out how I want to go forward in my life is, to me, a meaningful career will be a constant cycle between craftsmanship and passion, and each one will feed the other. Don't overthink which one you're prioritizing or which one goes first. Dedicating to your craft will bring you passion, and passion will make you dedicate to your craft. If you are stuck in lockdown or whatever's happening in your country, I hope everything's going well for you. And if you're being stuck at home and you have not much stuff to do, here are some authors that really helped me out trying to figure out what I want to do in my life. And I wish you all the best. Let me know if you want some specific recommendations from those guys.
Hi guys, I'm Alex again. I'm helping advise this year's iGEM team. I'm currently doing a PhD at UCL, so I've gone through a biochemistry undergraduate, rather convolutedly, I think. I originally applied to do, I left school with a sort of vague career aspiration. I wanted to do good for the environment. I wanted to fight climate change. I wanted to do something to sort of help restore, some of the wildlife. And I wanted to keep myself as open as I could, because I had no idea how to get there. So I had a sort of vague aspiration, but no clear steps. So I applied to do natural sciences. However, this wasn't as open as I thought it was going in. In fact, it was very closely sort of rooted into physics and maths, which I am more of a visual learner, and I like applications of things. So highly abstracted physics and mathematical applications, especially in the first couple of years where you're sort of, learning about condensed matter, and you're learning about the quantum fields of hydrogen. I felt like it just didn't have any sort of application to what I was interested in. However, I took a year out and I reapplied to do biochemistry, and biochemistry has a much more sort of visual aspect. It's, even though you're dealing on the molecular level, the end results and the protein structures are at least something that you can sort of see, visualize, and play with. Hence the squirrel, the revelation of biochemistry. It did feel like it at the time. I don't know if I could get a slide change. So what did I like and dislike about biochemistry? Biochemistry is literally understanding life. And I think that sometimes gets hidden in the sort of day-to-day learning about biochemistry, but that is fundamentally what it is. It's how chemistry becomes biology, and it's that interface. Sort of self-explanatory, I suppose, but it's sort of worth sort of maybe remembering once or twice, because I find that just incredible. And so the space in which biochemistry operates in is you go from the very small, the biomolecular, as already mentioned by the previous two speakers, very small proteins, nucleic acids and stuff. Then you go to the whole system type biology approach, whole organisms, whole tissues, and regulations on how you keep multicellular organisms and us alive. And I felt my physics background actually helped me understand some of the physical concepts in biology. So you can have interest in any sort of different level, and I think as a synthetic biology team doing this iGEM competition, you're probably seeing maybe slightly more weighted towards the bottom, the biomolecular level. There's quite a key concept in biochemistry is in synthetic biology is what you can't understand, you can't build. And obviously we're trying to build something. And so we're working from the bottom up. However, you can have many biochemical applications all the way through the different levels. In immunology, for instance, you have the whole drug effects and how the body reacts to certain drugs and how it's all regulated. But you also have, again, the smaller level within that. You have how ligands interact with individual proteins and stuff. And then similar to the others, the last thing, I also possibly, as I was more interested in the bottom level, I also personally disliked the cell signaling pathways. The higher up sort of level and how things link together, it's a bit beyond me, to be honest. I like to sort of focus and a bit more detail orientated. So I'll leave the systems biology to Ollie. So yeah, during my undergraduate, I was interested in a couple of things in particular, expanding off the previous slide. Firstly, I did a summer lab project on bacterial light harvesting complexes and reaction centers. So this is how energy, well, this is in bacteria and in plants, this is how energy, solar energy, enters into the biosphere and is converted into chemical energy, glucose. And what fascinated me is that you have these photosynthetic arrays in plants, which you can channel photons through to the reaction center, where you have the water oxidation reaction, the multi-stage water oxidation reaction, which then liberates electrons to feed into the Calvin cycle. But it's absolutely stunning, and this is a quantum coherent structure, which I think is pretty incredible. You have photons entering one side, and you have a resonant energy transfer across the reaction centers, which happens in the femtoseconds range without any loss of energy. And that's really incredible. Another thing that caught my interest during biochemistry underdegree was this fantastic researcher in MIT called Angela Belcher. She developed the virus battery, where they did protein manipulation on the surface coat of an M13 virus, which allowed it to bind common metal ions onto the surface. And it also allowed the head, if you will, of the virus to bind onto carbon nanotubes. And what was just incredible about this is that you had the same energy output from a virus battery as you did with a lithium ion battery. You had the same energy density, but with far less potential environmental degradation, as you don't have to mine rare earth materials to get it. And I thought this was incredible. You're manipulating on a nanoscale using your knowledge of the molecular basis of life, of nucleic acids. So my current research interests now, and my PhD project at UCL is working on things that look a bit like a virus-like particle, the virus capsule on which the virus DNA sits. The proteins in the protein, in fact, isn't actually a virus protein. It just looks and feels like one. But it's basically this protein shell, this hollow sphere, which I'm trying to modify the surface of to display potential cancer therapeutics, as well as putting enzymes into the interior to try and increase their stability for green chemistry applications. So trying to put enzymes in to increase their thermal stability so they can operate at slightly higher temperatures or for longer. Those are two issues at the moment in green chemistry. You want enzymes to operate for as long as you can, make it more economical. In terms of the cancer therapeutic, I'm trying to put biomarkers onto the surface. So as your cell becomes cancerous, you've got several checkpoint mutations, and those mutations are mirrored on the surface of your cell. And so by putting those mutated peptide fragments onto the surface of encapsulants of the proteins I work on, hopefully trying to train the body to have a better autoimmune response against those pre-cancerous cells. And as you can see, they're very small. I'm talking under 100 nanometers, about 40 nanometers, which is pretty cool. Again, working on the nanoscale using biology. So my career so far, after my my undergraduate degree, I was looking for a PhD. As I spent 5 years as undergraduate, I had no way to fund a master's in the UK system, at least, a master's degree from the using a government grant. So I was looking for a PhD opportunity, but in the meantime, I managed to get, with the university help, a internship at a scientific incubator, which exposed me to a lot of scientific companies on the early spin-out stages. Sorry, such as in Bristol, which was incredible to see, and it's definitely something that I'm interested in doing afterwards. Apart from that, finding a PhD, it took me the best part of a year, and I do feel like maybe having a master's background would have benefited, but got on in the end. And lastly, any advice? I think having as much, you can get a lot out of university. I think if you ask for it. Professors and lecturers are very accommodating. You can ask for summer opportunities. You can ask for, they generally have spaces in their labs, or they know people and they can put you in contact with people. Another thing I would say is joining societies. So in the UK, we have the Biochemical Society, and I'm sure we have other societies elsewhere in the world, which you become a member of as an undergraduate, and they will help fund summer opportunities, which really look good if you want to stay in the field and progress on to Masters and PhD, and in general, just for your general interest. Now, and I think that's my last slide, so I think we can take any questions, I think.